Hello students, we are going for the next heading called as the structure of TMV, tobacco mosaic virus. So this tobacco mosaic virus is nothing but the virus infects the tobacco leaf. The virus infects the tobacco leaf and causes mosaic disease. Hence it is called as tobacco mosaic virus. So, when you take a leaf or a tobacco leaf, on the leaf you can see there is a presence of white powder-like structures are being present on the leaf. So, here on a tobacco leaf, tobacco is called as Nicotiana tabacum. On the tobacco, there is a presence of white patches. White patches or powder-like structures are being present. Once white patches are being present on the leaf, automatically this areas the photosynthesis is not there. So it leads to loss of chlorophyll. Loss of chlorophyll is called chlorosis. So this is called as infected tobacco leaf. So, when you take infected tobacco leaf and see this under electron microscope, we can see there are many rod-shaped structures. So, the rod-shaped structures are nothing but TMV. I repeat this, TMV is a rod-shaped virus. Rod-shaped virus, and it is made up of rod-shaped virus made up of central hollow single-stranded RNA. Here there is a presence of single-stranded RNA is present. This is called single-stranded RNA as a genetic material. This single-stranded RNA has 6,600 nucleotides are being present. 6,600 nucleotides. The word nucleotide is nothing but it is the combination of nitrogen base, pentose sugar and phosphate. All these three together forms nucleotide. In single-stranded RNA, it has 6,600 nucleotides are being present. This is about genetic material. The genetic material is single-stranded hollow RNA is present. Single-stranded RNA is present as the genetic material. This single-stranded genetic material is covered by the protein units. There is a presence of protein coat is present, and this protein coat, the protein coat is called as capsid or capsule. So it is nothing but protein coat. So in one TMV, there is a presence of 2,130 capsomeres. 2,130 capsomeres. So what is capsomere means? The protein coat is made up of smaller subunits. Protein subunits. The protein coat is made up of smaller subunits, and the smaller subunits are called as capsomeres. In one TMV, 2,130 capsomeres are being present. This is even important for your NEET exam. Like this, there is a presence of a rod-shaped virus covered by the presence of a protein coat. So this is usually called as single-stranded RNA covered by a protein coat. Next, the length of the TMB. The length of TMB is, it is 3000 angstroms. The length of the rod-shaped virus TMB is 3000 angstroms. The diameter is 150 to 170 angstroms. This is called as the diameter. So the diameter of the TMB is 150 to 170 angstroms. So I repeat this. TMB is a rod-shaped virus, has a presence of single-stranded RNA with 6,600 nucleotides, and it is covered by protein coat. And this protein coat is made up of protein subunits, and protein subunits are called as capsomeres, and it has one TMB. One TMB has the presence of 2130 capsomeres with a diameter of 2 nanometers. So each one capsomere has a diameter of 2130 capsomeres with a diameter of 2 nanometers. So this is about the structure of bacteriophage. So this single-stranded RNA, as I said, TMB. When this TMB is enlarged, so at the center there is a presence of hollow single-stranded RNA, and this is covered by a protein coat. Here there is a presence of a protein coat, and this protein coat is made up of smaller subunits called as capsomeres. So all these subunits are called capsomeres. So this capsomere will have a diameter of 2 nanometers, and in one TMB there is a presence of 2300 capsomeres have been present. So this is called as the structure of TMB, structure of bacteriophage. Structure of TMB, tobacco mosaic virus. So we go for the next heading called as structure of bacteriophage. So the bacteriophage is nothing but it is a virus which infects bacteria. So the virus which infects bacteria is called as bacteriophage. This was discovered by Trott. Hence it is also called as T phages. It is called as T phages. So this bacteriophage has a shape of a tadpole or tadpole shape. It is being present with head and tail. Head and tail is being present. Hence it is usually called as a tadpole shape. Next, we go for the structure of bacteriophage. So the bacteriophage has a presence of polyhedral head is being present, or hexagonal head is being present, and this head is usually made up of 20 triangular facets of being present. Inside the head is made up of 20 triangular facets, and this is made up of protein coat, or there is a presence of protein coat. Inside the head there is a presence of double-stranded DNA. Inside this head there is a presence of double-stranded DNA has a genetic material. Double-stranded DNA has a genetic material. It is present inside the head. Next, the length of the head is 958 angstroms. Length of the head is 958 angstroms. Diameter of the head is 458 angstroms. But here in T4 phase, the length of the head should be equal to the length of the tail. So here the tail also has a length of 958 angstroms. The length of the head is 958 and the length of the tail is also 958 angstroms. Now we continue this. So here there is at the junction of a head and a tail, there is a presence of a collar. At the junction of head and tail, there is a presence of collar. Next, this head is followed by collar. Collar is followed by the tail. The tail is again covered by the protein coat. The tail is also covered by the protein coat and it has 20 rings of being present in the tail. The tail is also made up of protein coat. Next, at the end of the tail, there is a presence of basal plate, or it is called as tail plate, or it is called as end plate. End plate, tail plate, basal plate is being present. This basal plate has the presence of 6 tail fibers. 6 tail fibers are being present, and at the end there is a presence of 4 tail spikes are being present. This helps in fixation or anchorage to the bacteria cell. So this is nothing but the virus infects bacteria. So the bacteria cell is holded by the tail fibers and tail spikes, and it starts infecting the bacteria. So this is called as the structure of bacteriophage. Has head and tail, and this head and tail has the presence of 958 angstroms with 458 angstroms diameter and has a double stranded DNA. So one bacteriophage has a protein coat, and this protein coat is made up of 2000 capsules. The protein coat has the presence of protein subunits, and the protein subunits is 2000 capsules. So this is the structure of bacteriophage. So the tail has a presence of protein coat, and this protein coat has made up of protein subunits, and this protein subunits are called capsules. So the tail has the presence of 24 rings. It's not 20, it's 24. The tail has round about 144 capsules alone. As I said, 2000 capsules in a complete bacteriophage. Tail alone has a presence of 144 capsules are being present. There is a presence of 24 rings. Each ring, there is a presence of 6 capsules are being present to form 144 capsules in a tail. So this is about bacteriophage. So we go for the next heading called as viral diseases. So we know that the virus depends upon the host. They infect, they are classified into bacteriophages, cynophages, phycophages, mycophages, plantophages, or called as phytophages, as well as zoophages. Like this, based on the host they are infecting, it causes diseases in all. For example, it causes diseases in plants, animals, humans. In all, it is going to infect. So like this, we have various types of diseases. So we start with, first one is viral diseases in humans. Viral diseases in man. So AIDS is one of the diseases. AIDS, mumps, smallpox, herpes, influenza. So AIDS, mumps, smallpox, herpes, influenza are some of the viral diseases caused to the humans, man. Next, viral diseases in animals. So it causes rabies, mad dog bite, or we say that rabies. Next, foot and mouth disease in cattle. So it is nothing but foot and mouth disease in cattle. Next, distemper of dog. Distemper of dog. Next, encephalomyelitis of horse. Encephalomyelitis of horse. It is called as mad horse disease. So it is nothing but, as we use the word mad cow, here mad horse disease is being present, called as encephalomyelitis. Next, Newcastle disease in chicken. So this is again animal diseases. Now we go for diseases, viral diseases in plants. It is called as leaf rolling disease. So once the virus is infected, the leaves are curled, rolled. Hence it is called as leaf rolling disease. Next, yellowing of veins. So the veins, medullary veins and veinlets, all that becomes yellow in color. So the absorption of water food supply to the leaf will be less. Next, dwarfism or stunted. The plant never grows in height, they become dwarf. So it is called as dwarfism or dwarfing and stunted. Next, mosaic disease. So mosaic disease is nothing but there is a presence of patches on the leaf. Once there is a patches on the leaf, then usually the color change takes place. It may be white patches, yellow patches, red patches, like this. Different patches are there. So this leads to loss of chlorophyll. Once there is a loss of chlorophyll, no photosynthesis, the plant dries and dies. So loss of chlorophyll is called as chlorosis. So all these are nothing but some of the viral diseases. So we go for the next heading called as lichens. Lichens are nothing but it is a symbiotic association. Symbiotic association. Symbiotic means association. So it is a symbiotic plant or symbiotic association between algae and fungi. So here in a lichens there is an association of algae and fungi. So this is called as lichens. Now the algal association with the lichens, they are called as the algal association is called as phycobionts. Phycobionts. And the fungal association with lichens is called as mycobionts. So here algal association phycobionts, fungal association is called as mycobionts. Here the algal plant has a green pigment called as chlorophyll, so they help in photosynthesis. Here the algal plants undergo photosynthesis. This is nothing but preparing the food, manufacturing of food or preparation of food is called as photosynthesis. This helps the plant for supplying food. Whereas when you go for a fungal association, this fungi helps in absorption of water and the nutrients. Fungal helps in absorption of water and nutrients due to the algae. Algae association helps in photosynthesis. So algae acts as a phototrophs. Whereas fungi acts as a heterotrophs. So here it has both algal association as well as fungal association. Algae acts as a autotrophs, fungi acts as heterotrophs. It absorbs water and food due to the plants. So it is nothing but lichens. Very important point here is lichens are always called as pollution indicators. Because why we use this means when there is a presence of lichens or being grown, that area should be pollution free. When there is a pollution, that area the lichens never grow. So always they are called as pollution free areas. Lichens will grow, so they are called as pollution indicators. So we go for the next one. The lichens grows in different areas. So based on that, there are different types of it. The first one is lichens grow even in Arctic regions, and it is also growing in the winter climatic conditions. All these are called as reindeer mosses. One of this is called as Cladonia renifera. Cladonia renifera is called as radial masses. Next, lichens grows on the rock surfaces. Surfaces of the rock, it has been growing. They are called as sacchycoles. Sacchycoles. Next, lichens growing on the tree barks. Lichens growing on the tree barks is called as carpicoles. Carpicoles. Next, lichens growing on the soil. So it is called as terricoles. Terricoles. Like this, they can tolerate a very wide range of tolerance is present in the lichens. They can grow in all the climatic conditions, but it is polluted free areas. This lichens have been growing. Now we go for classifications of lichens. We'll go for synapses of biological classification. So overall synapses. First, we know that all the living organisms cannot be studied under a single heading. So we require classifications to study each organisms at different levels. So first, we are starting with Aristotle. We know that Aristotle is nothing but the father of biology, and he is also called as father of zoology, classified plants and animals. So in the plants, Aristotle classified the plants into herbs, shrubs and trees. He classified plants into herbs, that is small plants, shrubs, medium level, and trees are bigger one. Next, he classified the animals. He classified the animals based on the blood. So, anemia. So, anemia is nothing but the animals without blood. Animals which do not have blood are called as anemia. Anemia is nothing but the animals with blood. So animals which possess blood, they are called as anemia. Without blood is called as anemia. Like this, Aristotle classified the animals based on the nature of the blood. Next, we go for the next one, called as Carolus Linnaeus. So he is called as the father of taxonomy, and he given about binomial nomenclature. He classified all the living organisms into two kingdoms. So it is called as two-kingdom classification. Two-kingdom classification was proposed by Carolus Linnaeus, and he classified this into kingdom Plantae, kingdom Animae. So all the plants was included under one kingdom called as kingdom Plantae. All the animals was included under the next kingdom Animae. Next, we go for three-kingdom classification. Three-kingdom classification. This was proposed by Ernest Haeckel. So Ernest Haeckel classified three-kingdom classification. So the first kingdom is kingdom Protista, and the second one is kingdom Plantae and kingdom Animae. So this Haeckel classified all the living organisms into three kingdoms. Now we go for four-kingdom classification. So in a four-kingdom classification, this was proposed by Copeland. So this person classified into four kingdoms. All the living organisms was classified into four kingdoms. The first one is called as kingdom Monera, the second one is called as kingdom Protista, and the third one is called as kingdom Plantae. Plantae is also called as Metaphyta. Animae is also called as Metazoa. Now, in all this, one or the other was being left out. So finally, they accepted type of classification. The right type of classification is called as five-kingdom classification. So this five-kingdom classification is the right one, accepted one, and this was proposed by Robert H. Whittaker. R.H. Whittaker classified all the living organisms into five kingdoms. The first one is called as kingdom Monera, and the second one is called as kingdom Protista, and the third one is called as kingdom Mycota, and the fourth one is called as kingdom Plantae or Metaphyta, and the fifth kingdom is animal kingdom or Animae or Metazoa. So this is about classification, kingdom classifications. Now, when we step into each one, kingdom Monera. So in a kingdom Monera, all the primitive, microscopic, prokaryotic organisms comes under this. So here again kingdom Monera is classified into three types. One is called as Archaea. Archaea is also called as Archaebacteriae. So they live primitive, ancient, prokaryotic cells called as Archaebacteriae based on the habitats where they are present. Based on this, again it is classified into three types. One is called as methanogens. So these methanogens always live in extreme conditions in marshy areas. Marshy areas called as methanogens. The second one is called as thermoacidophils. So these thermoacidophils, they are present in the hot springs. They live in the hot springs. Halophils. So these bacteria live in the saline water or saline solutions or saline water. There is a presence of halophils. So this is about Archaea. Now when you come back to the eubacteria. Eubacteria is nothing but a true bacteria. So I am going to explain this later on. Next we go for cyanobacteria. So cyanobacteria is nothing but blue-green algae nostril comes under cyanobacteria. So this is about kingdom Monera. Now when we go back to bacteria. So bacteria is classified into different types. So we are talking about first we are talking about the shape of a bacteria. Based on the shape, there are different types. One is called as a spherical shaped bacteria called as cocci. Spherical shape called as cocci or coccus. Next, we go for the rod shaped bacteria. The rod shaped bacteria is called as bacilli or bacillus type of bacteria. The third one is called as comma shape. Comma is called as vibrio. Coil. Comma is called as vibrio. Coil shape is called as spirula or spirulum. So this is about based on the shape. Next, based on the locomotory structures, based on the flagella. Again, bacteria is of different types. First one is called as atritis. The meaning of atritis, here the bacteria do not have flagella. The flagella is totally absent. Example, cocci. Here the next one is monotritis. The word monotritis, there is a presence of single flagella towards one end, one polar end. There is a single bacteria, so it is called as monotritis. Now we go for lophotritis. So based on lophotritis, again only a tough flagella is present towards one end. The next one is amphitritis. The amphitritis is nothing but a tough flagella is present on both the sides. Both the sides there is a presence of tough flagella. Next, peritриtis. Peritриtis is nothing but a single flagella present all over the body. It is called as peritриtis. So this is about based on locomotory organs or flagella. Next, based on the cell wall, the bacteria cell wall has the presence of peptidoglycans, muramic acid and proteins have been present. Again, based on the mode of nutrition, there are two types, autotrophs. In autotrophs, photosynthetic, chemosynthetic. Heterotrophs, it is usually parasitic and saprophytic. Next, we go for algae. So in algae, there is a presence of different divisions of it present. The first division is called as Chrysophyta. Chrysophyta is the first division. The next one is Pyrophyta. Pyrophyta is the second one. Third division is called as Echinophyta. Fourth division is called as Myxophyta. And the fifth division is called as Protozoans. Again, protozoans based on the locomotory structures, again they are classified into different types. First one is amoeboid type. So here there is a presence of pseudopodia has a locomotory organ false foot. Whereas the second one is called as flagellated protozoans. In a flagellated protozoans, there is a presence of a single whip-like structure called as flagella, and this flagella helps in locomotion. Whereas the third one is called as ciliated protozoans. So in a ciliated protozoan, there is a presence of cilia as a locomotory organs. The last one is sporozoa. In sporozoa here, totally locomotory organs are absent. They do not have locomotory organs here. So when we go for the next one is fungi. Here in a fungi, it is a group of plants which cannot prepare their own food. It is called as achlorophyllus. So they cannot prepare their food, they depend upon other. This has 4 classes. The fungi, mycota has 4 classes. The class Phycomycetes. So they are called as Phycomycetes, or also called as algal fungi. The second class called as Ascomycetes. This is called as sac fungi. The third one is called as Bacteriophage, called as club fungi. Bacteriophage are called as club fungi, and the fourth one is Deuteromycetes. They are called as imperfect fungi. Hello students, we have completed the second chapter. Biological classification is being completed. So I have given you synopsis of the complete chapter. So go through all this, and from the next class we are going for neat classes. Prepare and be ready with the books. We are starting with objective type, multiple choice questions from the next class. So we start with the next one called as classifications of lichens. As I mentioned, it is an association between algae and fungi. The algal association is called as phycobionts, and the fungal association is called as mycobionts. So here, based on the surface, how the structure of the lichens are there, based on that it is classified into different types. The first one is called as crustose lichens. So this crustose lichens are always forms the crust on its surface. It is present on the rocky surface, or on the tree trunks surface they form crust, and it is usually present on the upper surface. Upper surface, they are will keep on growing, creeping, and it is usually attached. It looks like a rust. It looks like a rust, but here they are present on the surface. And here the example for crustose is gravis hematoma. This is called as the examples for crustose lichens. This will be in the form of crust on the surface of the bark, or on the outer surface of the plant bodies. It is being attached. So this is usually called as crustose lichens. Now, when we go for the next one called as foliose lichens. So in the foliose lichens, here the thallus is usually attached on the surface, and they look like leaf-like pieces of leaf-like flat, and the thallus will be lobed. Thallus will be lobed or irregular. Lobed or irregular margins are being present, and this is attached to the substratum. With the help of rhizines. Here there is a presence of rhizines. This rhizines holds the surface, and it is usually leaf-like pieces attached on the surface. So it is usually called as flat thallus, irregular shape, and this is usually called as foliose lichens. Example: Picea, Paramilis, Penticum. So all these are some of the examples of foliage lichens. Now when we go for the next one is fruticose lichens. In this fruticose lichens, it is well developed, or we use the word better organized thallus. Here on the substratum, it is usually grows on the host. With the help of the substratum, it is being branched. There is a presence of flat, cylindrical, it may be flat or cylindrical, narrow axis of being present, and it is being profusely branched and looks like a small plant hanging. Here, this hangings may be erect, or it is drooping down, or it may be in a various forms. It is being attached to the tree trunks. So it is usually pendular or filamentous. Like this, they are attached to the substratum. So it is usually profusely branched one. This is well developed lichens called as fruticose. Example: Usnea, Cladonia, Evernia. So all these are the examples of fruticose lichens. Students, we will go for the next assignment questions. For this chapter, I am giving you some more assignment questions. Explain, first question, explain the structure of bacteria. So bacteria structure, you are going to explain here with a labeled diagram. Now, we'll go for the second one. Explain the characters of kingdom Protista. So here, kingdom Protista characters should be explained here. Next, we'll go for name 5 divisions. 5 divisions of kingdom Protista. So in a kingdom Protista, there are 5 divisions. You are going to write that divisions. Next, explain the characters of kingdom Mycota. So in a Mycota, the general characters should be written here. Next, name 4 classes of kingdom Mycota. So in this, there is 4 classes are there. So that classes you are going to write here. Next, write a note on viroids and prions. So you are going to write what are viroids, what are prions, and what is the diseases caused here. Next, what is CJD? So you are going to explain and write the note on it. So this is what is CJD. Next, write the living and non-living characters of virus. So living and non-living properties of the virus should be mentioned here. Next, draw with a labeled diagram of TMD. So here you are writing the rod-shaped diagram as well as along with that the section or portion enlarged along with that. Next one is bacteriophage diagram to be written and labeled. Next, classify, write the classification of lichens or lichens. So here there are 3 classes are there. You are going to write the classes with examples to be written.